the transport of pollutants. We considered wind speed, humidity, and temperature. And from that reference point, we also figured out a second experiment. And here we had 2 sensor nodes. One was moved directionally from the central sensor node in approximated intervals, collecting a similar set of data set. And the other one was moved randomly around the central sensor node. Sometimes it goes beyond the intervals fixed sensor node, and so on. So we captured a huge data set that we dealt with in training our machine models. To train the machine learning models, it was not just to wake up and think of the best to pick any machine learning model for use. We did the research in relevance to this field of study, and we got that some machine learning models have been projected as best performing models for this application. So we chose 4: LSTM, ANN, SVR, and random forest. And using the data that we have cleaned, we trained these models with a number of features. PM in the 3 scales, weather parameters, 3 parameters, wind, humidity, and temperature. Then we gave it location, and we also introduced another parameter where we computed Euclidean distances. So we could rely on these parameters to predict the 3 PM scales. The performance of the models is here. The R scores are very high, and the other measures are quite low, showing the importance of the application of the models in our study. And the chart here shows what we expected if we employ these models in our real-life experiments. Inferring the spatial distribution, we did the experiment following the weather parameters, and wind speed turns out to be the best in influencing the spatial distribution of these parameters regarding the prediction accuracy of the models that we developed. So we have this performance for concentration, prediction accuracy versus Euclidean distance first, without any weather parameter. That was an important feature for us. Then we compare the performance when we introduce wind speed, starting from the right of that graph. And when you look at the graphs carefully, you can see in the labels, SVR is seemingly doing better than the 4 other machine learning models that we projected. We also did the same when the weather parameters, all of them are combined against the PM concentrations. We obtained those results. And because it could appear difficult to make inferences from here, we said, well, let's plot the prediction accuracies for each spatial position that a number of samples were plotted. And here now you can see clearly for the level we have gotten to, SVR is doing better than the other machine learning models. So that was kind of the experiments we did. And in conclusion, as of now, we have chosen SVR model as the best model for our application. And for these reasons, the study allows us to make to be able to track and localized pollution sources and be able to talk about the influence of weather parameters in this application, a feature that has been omitted in the existing studies.
Okay, good afternoon. Okay, I'm Lawrence Kinoti, and I'm here to present my findings on my study on the logging moratorium in Kenya and its impact on Community Forest Association, whereby we took a case study of Meru County State Forest, and my supervisors were Professor Moria and Dr. Hilda Omae. And maybe to start with, logging moratorium is basically the ban on logging activities in the state forest. Okay, it is a banning of logging activities in the state forest. And forest sector is a very key sector in our social and economic activities in Kenya as a country, because we get most of the resources economically from the natural resources, so that formed the basic interest of this study. And again, when you talk of the climate change aspect, there is the issue on whatever happens in the forest in terms of the nature-based solution in line with climate change is very of much interest in climate change studies. So, the Forest Act of 2016 provides for the decentralization of the forest management to the adjacent communities through the CFAs. The CFAs are basically the community forest associations. And what motivated our study of the research problem as per se is to get the impact on the logging moratorium. It is vital to find out what drives the forest and adjacent communities in forest activities. And when you get that solution, then you can be able to tell what has been the impact on the logging moratorium. Okay, a study by Mutune et al. 2015 on local participation in CFA at Sururu and Eburu Forest. It was established that households participated in CFAs to derive livelihoods gains, and that unclear participation benefit offered little incentives to get involved. The general objective was to determine the relationship between logging moratorium and CFAs in Meru County, with specific objectives being to rank forest use and benefit, number one. Number two objective was to determine the impact of logging moratorium on CFAs livelihood, and finally, to establish the impact of logging moratorium on CFAs effectiveness. In line with the scope and sampling design, data analysis and limitations, purposive sampling was used to select the 2 CFAs to be used as a representation of Kenya state forests, others excluded due to time limits and also the fund constraint. The study entailed a data sample of 289 respondents from the 2 CFAs picked randomly from the symbios of the forest and adjacent communities, and the 2 CFAs were Rodombe CFA in Saudimenti and Mutiene in Buri. Then the primary data was obtained from the pre-tested questionnaires, focused discussion group, and key informant interviews to help achieve all the objectives. Secondary data was instrumental in achieving the second and the third objective, whereas quantitative data from the household survey was organized in SPSS to get descriptive statistics on mean and percentages used in the analysis of the objective. Tables and figures used dominantly in my data presentation. When trying to determine the first objective on the most preferred use and benefit of the forest, I find out that PELIS, which is Plantation Establishment and Livelihood Improvement Scheme, whereby you invite the community to plant trees as they grow their food crops, ranked. It was the most preferred use and benefit, followed by beekeeping, which is an indirect benefit of the trees. Then firewood came that, timber, then medicinal collector was the last. In terms of those that take part into my study, women took part largely in the study at 56.7%, and men followed at 43.7% of the respondents of those interviewed, whereas 86.2% were the decision makers, and we were interested in finding the decision makers in the family, so that at least we can know if they qualify enough to give us the opinion, because you cannot form an opinion if you are not a key decision maker. Then on the size and livelihoods, tenure system of land, and I've talked about the most preferred use and benefits. We found that those with no land, out of the 289 respondents, 29.8 didn't own any land. And those that owned at least 0.1 to 1 acre, they were 42.9. Then 1.1 acres to 2.9, they were 19.7. Then 3 acres to 4.5, 6.2. Then those with 5 acres and above, 1.4. And that now poses also another interesting line of study, whereby the subdivision of land, you can see, is very dominant in our area, which also might affect food security in the long run. In terms of the tenure system, those with no land, they are 28.7. Those that have inherited the land that they have, 68.9. Then those that have rented land, 1.0. Those that have bought, 1.4%. Then, on being asked some specific question that I will read through, 93.4% believe that legal logging promotes job creation in the sawmill value chain. Then 94.1% believe that employment creation and direct benefits empowers CFAs economically. Then 82.7% believe that by forest and adjacent communities securing goods, they do not destruct the forest. Then 96.1% believe that a well-conserved forest is an indicator of economically empowered community, with 79.9% believing that with IGAs there is minimal illegal logging. Maybe I can go to discussion. Forest user benefit on ranking Pelis was top, beekeeping livelihood collection. The implication is that forest conservation cannot be delicted from livelihood improvement by FAC. Then closure of sawmills resulted into retail business collapse due to reducing purchasing power. Then the impact of logging moratorium on CFA's livelihood, 29.8% don't own land, implying that they rely on casual labor, and leasing land to support livelihoods by scaling down independent sectors will give a negative impact on the Big Four agenda. And on domestic fuel, 87% depend on firewood. Supply is from the private forest and public forests. Moratorium resulted into the scarcity, thus high prices and reduction in the marginal propensity to save. And since I've talked more of the findings, the discussion, let me just go through the conclusion. The study recommends that the Cabinet, Secretary of the Ministry of Environment and Natural Resources, and Forestry should ensure that environmental regulations put in place should not delink forest conservation from livelihood improvement by the forest and adjacent communities. And then the study also recommends that the government should adopt a regulation mechanism that safeguards logging on extension forests of the community whenever there is an ongoing logging moratorium in the public gazetted forest. Also, there is need to strengthen the CFAs and also have policies and registration, and government action should have qualitative assessment indicating changes in ecosystem health in order to establish effectiveness of policies and actions. Thank you.
Do justice to these presentations. Thank you.
Good afternoon, everyone. Okay, I'm Purity Wanja, presenting a topic on drivers of water pollution of Kuru River in Meru County. And my supervisors are Professor John Moria and Dr. Cynthia Mugo. On to my introduction. Water pollution is a concern because it is affecting the quality and availability of water resources essential for various uses in our households and even at our farm level. Kuru River is a tributary of Tana River that serves as a source of drinking water, water for irrigation, and other domestic uses within Meru County. And this is as per the subcatchment management plan that was done in 2016. The river flows through an area that is densely populated, and the main activity within the stand area is mainly agriculture and livestock rearing. And this also is as per the subcatchment management 2016. Problem statement, according to UNEP, in the year 2004, 40% of the total population will live in water-scant area. That is by the year 2025. That is like 2 years from now. Kenya water resources are progressively getting polluted from both the point and non-point sources due to agriculture, industrial and domestic waste, and also leakages from the pit latrines, which are mainly used in our rural areas. Kuru River is prone to contamination due to the dense population and the increased anthropogenic activities in the Standy area. Justification to this study: Kuru water resource has faced considerable levels of pollution, and due to water demand that is rising from the population growth within the area, and also the agricultural activities that are mainly, that is the main economic for the population or for the within the Standy area. Improved water quality for human consumption and sustainable ecosystem services and goods that are associated with the resource. So, due to need for water quality, there's a need to study to do this study so that we're able to get or to know on how water quality in Kuru River can be improved. And also, there is a scanty information about the quality of water in Kuru River. Research questions: how does the water quality of Kuru River vary from the source to the major tributary, that is Kadita River? And the source of Kuru is just behind the university, that is in the Loimenti forest that we're able to see in the morning. The second question: how have anthropogenic activities in the riparian zone affected the quality of the water of Kuru River from the source to Kadita? And the third one: how have the existing legislative framework and institutional regulators contributed to the conservation of the river? Objective: the general one is to assess the drivers of water pollution in tributaries of the Apatana River catchment within the Kuru River. Then specific objective: the first one is to quantify the physical-chemical properties of water of Kuru River from source to Kadita. The second, to determine the land use and the environmental conservation activities in the riparian zone of Kuru River from source to Kadita. And the third one is to evaluate the contribution of institutions provided by the legislative framework to conserve the water. The research methodology: I collected the water samples from 9 sampling points from the source to where the water or the Kuru joins Kadita. This water was to determine the physical-chemical parameters in the water, and that was pH, turbidity, conductivity, temperature, total dissolved solvents, nitrates, and phosphates. Then I also had a semi-structured questionnaire that was administered to 384 households along the river. And then also, there was also another semi-structured questionnaire for the institutions that are mandated in the conservation of the river. Those are the results showing physical-chemical properties of the river along, those are the 9 sampling points. And then these results were now compared to the standards that have been provided by various institutions, like WHO, NEMA, and KEPS, and United States of Environmental Agency. The results are like shown. Then from the same structure and questionnaires, we can see the blue one, the blue color, it's showing that 75% had no idea or knowledge on riparian conservation, while only the 24.87% had a knowledge on riparian conservation. Then on the graph, from on the far end, it's an involvement in riparian conservation. The people along the river that have been involved in the conservation of the river, and the bigger one is 62% that said yes, they have been involved in conservation. Well, the 37 says that they have not been involved in the conservation of the river. So this can tell us how this river has been conserved, and if the riparian conservation is not conserved, then pollution may occur from different points. This is community awareness of participation in the conservation of the riparian areas and involvement of institutions, regulators. We can see 44% are not aware of the laws that allow them to participate in the conservation. Then the source of information, where they get information on these laws, we can see 29 get from the RUAs, 52 from the Symbios, then we have 9 who get from others. Then opinion on the community awareness on the law, yes says they are aware of the laws, then 63 says they are not. Management plan of the Kuru River, 30% says they know that there is a management plan, while 74% are not aware of the management plan. Involvement in decision making by the RUA, only 23 have been involved by the RUA. Then county involvement in conservation of Kuru River. The community along the river are not aware if county government is opposed or is mandated in conservation of Kuru River. Then conclusions and recommendation. Anthropogenic activities in these are the 3 sampling points amongst the 9. So the anthropogenic activities in these sampling points might be the major drivers of pollution in Kuru River, whereby most of it's agricultural and livestock keeping, and even mostly taking animals to drink water from the river. And then there was lack of awareness regarding riparian conservation attributed to insufficient stakeholder involvement. Most of the stakeholders from NEMA, county government, KFS, all of them, most of the people interviewed were not aware of this, and even these stakeholders themselves says that they have not been involved because of various reasons like funding. Periodic assessment of the water quality in the Kuru River is recommended so that we're able to know the pollutants that are deteriorating the water quality. Then effective stakeholder involvement to raise awareness of riparian conservation is also essential. Thank you very much for listening.
Good evening, all. Yes, this is Victor Kimboy, a second year master's student taking master's in horticulture in the School of Agriculture and Food Science. So, welcome to my presentation. I'll be presenting on the effects of land preparation method and organic soil amendment on soil properties, growth, and yield of maize. Yes, since we know that maize is a staple crop, food crop in Kenya, and from the first start 2021, the production of maize in Kenya is about 3.57 million metric tons per annum. And since I touched on my title that there is land preparation method, the types of land preparation method that I'm using is two. That is the conservational land preparation method and conventional land preparation method, where the conservational land preparation method are measuring on the minimal soil disturbance. And the other thing that I have touched is about the organic amendment. And in my study, I'm working on the biochar, that is the organic substance rich in carbon. Another one is black soldier fly manure or frass fertilizers, that is from the sanitation project. And another one is the trichodama, that is the mycorrhiza fungus, which is inoculated into the soil. My objective was to determine the effects of land preparation method and organic soil amendment on soil physical property and maize growth and yield. And my hypothesis is that land preparation method and organic soil amendment has an effect on soil physical property and maize growth and yield. The problem which I'm addressing here is that there is an issue of soil health degradation, and that has contributed to a decline in food crop production, but here I'm measuring on maize crop production, which has declined here in Kenya. My methodology, the experiment I conducted here at Meru University, demonstration farm, and the layout of the experiment was in split plot design with a randomized complete block design, where for the split, where I had here, I had two land preparation method. I have one land preparation method with a pacing method. I didn't till the land, but the other one here is a conventional land preparation method, which is tilled. Normally, you do primary cultivation and secondary cultivation, and you do leveling. But this one, you just prepare the holes for planting. And I replicated 3 times. And on the data collection, I took the data collection for soil moisture using the tension meter or the soil moisture meter. For the soil bulk density, I used the coring method, that is there on the other end. And for the spud readings, that is the greenness of the leaves, I used the chlorophyll spud meter. And for the yields, I used the weighing balance there you're seeing to measure the yields per unit area. One of my findings is that these were my treatments. I had 2 land preparation method, that is the conservational and conventional land preparation method. I had about 10 treatments with different rates of application per hectare. I had 2.5 tons frass fertilizer, 5.5 tons, and 8.5 tons to compare their effects on the production of maize, varying the quantities. And again, to check the effects of biochar, I took the 2.5 tons and 5.5 tons, amended with the biochar to check the effects of the biochar on the soil moisture and the soil bulk density, and if it has an influence on the growth. Again, to check the impact of trichoderma, I took the 2.5 and 5.5 again, amend with the trichoderma to check if 2.5 tons PSF trichoderma has an impact compared to the 2.5 PSF alone. That is the first fertilizer. And here I had the control without anything, and I had the DAP plus CN fertilizer, which farmers are using. Because as you can see from these 5.5 tons farmyard manure, up there, those are the pure organic. But here we have the chemical fertilizer, which is normally used by the farmers. The reason why I introduced the farmyard manure here is that it is commonly known by the farmers. So I was using as a positive control. So the three here are the controls: farmyard manure, DAP, and this is the chemical fertilizer and the control to check. So this is the data for the moisture, and I found that the conservational land preparation method recorded the highest moisture percentages throughout the growing season, and this is due to reduced soil moisture. This is due to reduced runoff and increased water infiltration and exposure to sun for evaporation. Because as you can see, the conventional recorded the lowest as compared to the conservational. For the case of nutrient levels, the one which recorded the highest moisture level is the one amended with the 5.5 amended with the biochar. Because as we increase the biochar, we are increasing the amounts of the organic matter into the soil, which has a high hydrophilic properties to hold moisture. I think I've explained on that. Another parameter was on soil bulk density. When you compare the land preparation method, the two, they had no significant difference for the case of soil bulk density, but on the application of different nutrient levels, the one amended with the biochar recorded the lowest soil bulk density up to 0.6 grams per centimeter cube. And the control recorded the highest up to 0.9 grams per centimeter cube. I've explained that. The other one on the chlorophyll index, the conservational and conventional land preparation method had no significance difference for the chlorophyll, but on the application of different sources of the different rates of frost fertilizers, the one with 8.5 recorded the highest, followed by 5.5 plus trichoderma, followed by 5.5 PSF plus biochar, followed by 5.5, and the one which recorded the least was the control. But here, when you compare the chemical, which is normally used by the farmers, compared with the 8.5 tons frost fertilizer, it's somehow the same, because this one is a pure organic and this one is chemical. And the more we induce chemicals into the soil, the more we destroy the soil microorganisms. I'll explain that. On the yield, conservational land preparation method recorded the highest, followed by the conventional. And for the case of different manure levels, the ones with DAP plus CN recorded the highest because the nutrients was instant compared with the others for the organic nutrient levels. So after the DAP, then the one which followed was 8.5 tons PSF, because as you increase the amounts of BSF, you are increasing the nutrient availability into the soil. Sorry. The ones which recorded the least is the control, because there was no any nutrient applied. In my conclusion is that conservational agriculture or conservation land preparation method recorded the highest yield of maize per meter square. And on application of biochar, it reduced the bulk density up to It reduced the bulk density up to 0.6. Yes, thank you.
We have heard you. Thank you. Let's appreciate him. Next. Again, questions at the end, please. All the questions, hard and easy, all of them.
Good evening, everyone. Good evening, everyone. Can you hear me? I am June Sharon Kinoti, a current master's student pursuing Master of Science in Entomology, and I'll be presenting my concept note on the potential of the black soldier fly Hamicia illucens larvae to convert avocado organic waste into organic fertilizer. A quick background. I'm sure we are all familiar with avocado. It belongs to the family Lauraceae and to the genus Passia. It originates from the Central America and has widely been planted all over the world. In Kenya, there are more than 40 varieties of avocados, and according to Asila et al., Hass avocado is the main exporter variety, and Fuerte is preferred for processing. So, during a time of glut, when farm gate prices are low, a lot of fruit goes to waste. And this is because they have a very short lifespan, so they go bad easily. That's what I would say. So a lot of organic waste is left to decompose, and it can produce methane, which is a greenhouse gas that contributes to climate change. So we are actually looking for a solution to such issues. So avocado as a popular fruit has a high nutritional value, and is of economic importance. The production and processing of avocados generates significant amounts of organic wastes, including fruit pulp, skins, overripe avocados, as well as leaves. As you can see, these are some pictures that I've taken of avocado leaves as waste, which is organic waste, and what you'd expect to see in an avocado farm, and anything else that would actually go to waste, because an overripe avocado becomes organic waste, which is where the black soldier fly comes in. They are known for their ability to efficiently convert organic waste into nutrient-rich frass, which is a natural and sustainable alternative to chemical fertilizers. They are able to decompose and break down organic substrates and returning nutrients to their soil. They actually break down about 96% of all organic waste. So this study aims to investigate the feasibility of using the black soldier fly larvae to convert avocado organic waste into organic fertilizer. This is the life cycle of the black soldier fly, with about 320 eggs that hatch in 4 days. They go to the larval stage, 10 to 52 days, where they feed constantly, which includes 5 instars. Then they go to the prepupal stage, 7 to 10 days when they're not feeding, and they migrate to a dry site. And then they go to the pupal stage, which has a minimum of 8 days where they're not feeding and not moving. Then they go into the adult stage, 8 to 9 days, where they mate and lay the eggs. Why did we choose the black soldier fly? They have an efficient digestive system and are able to consume and break down a wide range of organic waste materials, with the digestive enzymes helping in the breaking down of complex organic compounds and facilitating nutrient release. They have a high nutrient conversion efficiency, where they are able to assimilate nutrients from the waste material and actually bring it back as what would actually be available to the plant. Also for their frass composition, which is waste that they give after they've already fed on the organic waste, it contains a balanced ratio of macronutrients and micronutrients. So macronutrients are the iron, zinc, and manganese. That's micronutrient, and macronutrients is nitrogen, phosphorus, and potassium. So a problem statement. In 2017, according to Wasilo et al., out of the 81,000 tons of avocados produced in Kenya, 30 to 40 tons went to waste due to pre- and post-harvest handling practices, which means half of everything that was collected actually went to waste. So this is a large amount of organic waste, and not counting the leaves in the shamba or any other waste that we cannot actually quantify at this particular point. So a lot of waste is seen. And also another issue in the fact that we have been overusing our farms, and we actually have developed soil infertility problems, and this requires us to constantly use fertilizer applications. So by actually being able to make a fertilizer, we'll be able to solve these two issues, the amount of waste and as well as having a fertilizer that is organic. My research questions are, how efficient are black soldier fly larvae in converting avocado organic waste into organic fertilizer? And what is the optimal waste to larvae conversion ratio? What is the nutrient composition of avocado organic waste? And how does it vary among different parts of the avocado plant? And the third one is, what is the nutrient content and quality of the organic fertilizer produced from black soldier fly larvae fed with avocado organic waste? And how does it compare to other commercially available organic fertilizers? My research objectives are, the general objective, to investigate the potential of the black soldier fly larvae to recycle different avocado organic waste streams into competent organic fertilizer, the specific ones being to determine the conversion rates of black soldier fly larvae of avocado pulp, peel, seed, and leaf, to analyze the nutrient content of the resulting frass as organic fertilizer, and to assess the effectiveness of the BSFL frass organic fertilizer compared to biocam. My justification. This particular project will offer a sustainable waste management project. By converting the waste into organic fertilizers, we address a challenge of waste management and promote a sustainable solution to reduce landfill. We'll be able to efficiently use resources by maximizing the utilization, by harnessing what would have been waste into something that is actually useful. And it also enhances the soil fertility and plant nutrition, because they're able to actually get these nutrients from what would have been waste and has actually been turned into a fertilizer. Also, it reduces the environmental impact on what would have been pollution otherwise, and there's economic viability because it creates opportunities for waste management, insect farming enterprises, and organic fertilizer producers. My methodology will be collecting and characterization of avocado organic waste samples, rearing of the black soldier fly larvae in a controlled lab, which we'll get from the rearing unit at Meru University, the monitoring the growth parameters and the nutrient contents of the black soldier fly larvae, harvesting and processing of the black soldier fly larvae frass to obtain organic fertilizer, conducting greenhouse experiments to evaluate the effect of the black soldier fly frass fertilizer, and analyzing the nutrient composition and quality of avocado organic waste black soldier fly larvae and the black soldier fly frass fertilizer using standard laboratory techniques. My expected outcomes are determination of nutrient composition and potential of avocado organic waste as a substrate for black soldier fly larvae, insight into the growth, performance, and nutrient content of the black soldier fly larvae reared, and the evaluation of the nutrient composition and quality of the organic fertilizer produced from the BSF frass. So some of the references. Thank you.
And you don't have your presentation, can I see by a show of hand? No one, so we are good.
Thank you. Good evening. I'm Jared Gisore, master's student, food science. And my project is on processing and characterization of camel milk butter. My supervisor is Professor Joshua Arimi. To start with, we know of camels. They mostly live in desert or arid and semi-arid areas, and they are mostly used as a source of food or transport. Traditionally, milk was consumed either fresh or in sour form due to the difficulties which were involved when processing some of the products. Then camel milk butter was produced traditionally from sour milk because there was insufficient milk to process a sweet cream butter. Guts and goat skin were used in terms of processing of the butter.
diameter. As we can look at the values, we can easily conclude that when the tomato that grew under normal condition and those that grew under Wambugia ugandensis stem bark obtained crude extract using dichloromethane, their values had no significant value. So we can just, when we use plant extract, like for Wambugia ugandensis, we can conclude saying that it influenced the growth factors despite being inoculated with the Rastonia soranceram. Now, this is the number of fruits. If we can observe the data there, we will, under normal condition, and those tomatoes that we grew under Wambugia ugandensis extract using the bark obtained from using solvents like ethanol and dichloromethane, that at the end of the day, their mean number of fruits was the same. Conclusion: inhibition zone of Wambugia ugandensis stem bark and leaf against organic solvents extracts obtained were significant. All the crude extracts obtained from Wambugia ugandensis were inhibitive against Rastonia, and then Wambugia ugandensis stem bark and leaf extract reduced the bacterial wheat disease incidence and severity.
Thank you, Oliver. Thank you. Let's appreciate him. Okay, the last presenter, welcome. Call you later. I'm in the meeting.
Oh, sorry.
Yeah. Hello. Good evening. I'm here to bring you a bit of cheer, so that at the end of the day, can go home and say cheers. We always have to wait for happiness. Let's wait a bit. Are we winning? Are we winning? Nakupea.
Umekubali, eh? Okay.
So finally, technology, right? This is how Baba lost his votes. Eh? So hello again. My name is Pauline Mwangi, MSc Entomology. I prepared slides on my concept note on the potency of chia plant crude extract in the control of cowpea weevil, also known as Colossobruchus maculatus. It's in the order Coleoptera, family Chrysomelidae. Into introduction. So cowpea is an important legume in Africa, but despite the importance of the crop, infestation by Colossobruchus maculatus has been a major constraint, hindering its large-scale production and storage. So bruchids, which is another name for Colossobruchus maculatus, also known as cowpea weevil or cowpea beetle, words which I'll use interchangeably in this presentation, is a major pest in stored cowpeas, especially in the tropics where conditions of high temperature and relative humidity favor it. Infestation may begin in the field, but is more devastating or destructive in storage, and left unchecked, some have reported up to 50% of loss of harvested cowpeas. So damage by bruchids results in grain loss, reducing the degree of usefulness, and making the cowpea seeds unfit for human consumption. And that causes rejection of farmer cowpea in export market trade. We all know that export markets, the standards are higher in terms of the quality. So if you have infected cowpea, you can't really subject it to export. So this has reduced or has had a negative effect on farmer incomes as well on the state richness. Because of course, when you export, you get external revenue or revenue exchange. So it is estimated that the economic losses caused due to Colossobruchus maculatus infestation in stored grain legumes, for example, in Kenya, that is in 2009, was at 73%. So that's very high. So on to biology, still under introduction. The cowpea weevil is a holometabolic insect. Of course, it goes through the 4 stages of egg, larva, pupa, and adult, with the female laying a minimum of 20 to a maximum of 40 eggs, of course, under optimum condition. Each egg can develop into a reproductive active adult in 3 weeks. So eggs are transparent and oval and are glued onto the seed surface. And once hatched, the larva bores into the seed, undergoes 4 molts and pupates inside the seed, and the adult is what emerges. So the adult matures 36 hours after emergence and are alive for 14 days. However, they do not feed, as their work is just to mate and oviposit in the case of females. So here we have a pictorial slide. It's not very clear, maybe because of technology as well. So the first slide basically was to show us the difference morphologically between the male and the female. We had another one over there of the life cycle, of which we have gone through also. And the bottom part is to show us the damage we should expect from brookids. So we go to the wonder plant, the sleeping beauty, chia plant. So chia, also known as Salvia hispanica linnaeus, is a well-known plant. And it's basically classified in the mint family. It is a flowering plant. The flowers can either be white or violet. It has been reported that chia leaves consist of various essential oils that have a potent repellent property, and they protect the plant from many insects, which is actually what we want to explore in this investigation. Although it's not only the leaves, you're going to subject other parts like the root bark. So on to the problem statement. Cowpeas are widely grown in Kenya, especially in medium to low altitude areas. It is considered a poor man's protein because, of course, of the cost. It is affordable, it is cheap. It is, however, difficult to store due to brookids, which cause heavy losses in terms of weight loss, germinability loss, quality loss. So the female cowpea brookid, to add salt to injury, we talked about how many eggs she can lay. The development cycle takes 3 weeks. So considering we usually store for long term, you can imagine the population that will be there in the end. So under justification, the most widely used method of control of not only Callosobruchus maculatus, but other storage pests is the use of inorganic insecticides, which apart from being inorganic, they also have a very long residual effect. So this is, however, a growing concern. There is, however, a growing concern over such use, as more and more people are preferring more organic products, more biodegradable, more safe to the environment, less toxic to mammals. As we are listening to VC in the morning, he talked about Roundup. So some of these chemicals are, when misused, improperly used, and there's all this hullabaloo about Roundup. I don't want to go into it. But a need has been created for more organic and safe-to-use alternatives, such as botanicals. Not only that, but organic insecticides, such as crude extracts, or for example, the chia plant, save costs and are readily available even at critical times of need, such as during harvest and storage, because they are not subjected to importation. Under objectives, general objective is to evaluate the potency of chia plant parts, crude extracts in the control of brookids. Under the specific objectives, to determine the lethal concentration under ambient temperatures and relative humidity, to compare which chia plant part has the highest efficacy, and to determine the effect of the crude extracts on oviposition and adult emergence in brookids. So under methodology, to be at 2 tiers, because you have the part of the cowpea brookid, whereby we have to rear them under optimum conditions, relative humidity and temperature in the lab. Then the second part will be about the chia plant, collection of samples, and of course extraction of the extracts. So chia plant extraction will be done based on established procedures after drying, as in Loni and Panahi in 2015, whereby after drying, they were subjected to hydrodistillation. Hydrodistillation provided the one of those days, right? Everything doesn't work. Eh? So the chia, of course, once hydrodistillation has occurred, we remove the excess water using anhydrous sodium sulfate, and we are left with our extract. So then comes on the next phase of our method, which is actually subjecting the cowpea brookids, which have already been read, to the extracts. So in this process, for us to establish the lethal concentration, that 50 should be a subscript, I'm sorry. So the crude extract of the leaves, the root bark, and we're also thinking about using the seeds, will be applied directly on the bruchids to establish the lethal concentration under controlled temperatures and relative humidity. So the concentrations will vary based on the procedures. And then for the oviposition, once we have our extracts and we have our specimen on which the cowpeas have laid their eggs, we'll subject them to exposure of the extracts under controlled environments in glass vials, sealed, completely sealed, with filter papers soaked with the extracts and sealed inside inside the glass vials with the cowpea seeds containing the oviposited eggs. So that's basically the method. Then these are the references we used, and I thank you.
Thank you, Pauline. And thank you all the presenters. We've done all the presentations for the afternoon session. And now we need to give some constructive feedback for all these presentations. I know time is a bit gone, but we owe it to our friends to give very constructive questions and comments. That's why they came to present. So what you're going to do, you'll just show me if you have any question for any one of them, and then mention to whom you are asking, yeah? We won't come back to you. Just mention whatever questions you have and mention to whom they are directed. Okay? Joker? Get your... Please note the questions that are directed to you, please.
A question for Victor. Victor for black soldier fly. Is it economically viable for the farmer to for example, for biochar? Because you know biochar is not easy to make. Is it viable for the farmer? And the second thing, also for the black soldier fly, how much should the farmer also give or use to produce enough black soldier fly also to help in the development of the organic matter? Also for the black soldier fly, I was thinking in this case where we have insects or pests, we're having insects being grown away from probably where they're being processed, and they go outside the development process. Could it be a risk where they could have a genetic makeup and produce, probably it become a pest or become a problem in the near future? Thank you.
Thank you, Moses. 2 questions for different people. Yes?
Thank you so much. First, it's a comment. Most of the slides that have been displayed, I don't think they are PowerPoints, they are power sentences. So I indulge the presenters to check on that, because it's a PowerPoint, it's not a power sentence. Then there's somebody who presented about organic, conventional and conservative types of farming. So my question is this to that person, I can't remember the name. What makes virgin lands more productive than non-virgin lands that have been left to idle for some time? What causes the difference? Then I have a question to, I think it's Sharon Kinoti, on avocados. Is there a difference between the organic avocados or the newer versions of avocados when compared to the indigenous avocados, the ones that grow tall and huge? Is there a difference on the same? Then on those who are presented on chia and BSF, yes, these researchers I have heard about these researchers for some time. why did they choose those particular, like now the BSF? Why choose BSF and not any other types of houseflies? Why? Because I think we are likely to cause monotony in a small area. Thank you.
Thank you for your questions. And I hope you have noted he didn't have names for some of the questions, but I hope everybody has noted which question belongs to them.
Again, I will not remember names for everyone. There was the gentleman who was growing maize. Oh, that's Victor, yes. Victor, what was the role of trichodama in the treatment? Because I had you use a name that I'm not sure. Did you mention mycorrhiza? Then please explain what is the role of trichodama and why trichodama and not in other fungi that you used. Number two, you said farmers tend to use DAP. I will actually disagree. For maize growing, most farmers tend to use NPK. DAP, especially in this region, DAP is used for other things. Most farmers use NPK for maize growing. So I don't know why you decided to use DAP. So please, you can, maybe I'm wrong, I could be wrong, so you can correct me if I'm wrong. Madam Kenoti, first, I know you've been asked a question. There's someone who said the bigger indigenous avocados. I want to correct them. We don't have indigenous avocados in Africa. All avocados are introduced from Central and South America. But I get your question. You're talking about the different varieties, the older varieties vis-a-vis the newer varieties. But I would want to ask Kinoti, why decide to, I'm trying to look for where the question I wrote, to biodegrade rather than improve the output of avocados. Why not develop methods of extracting avocado oils rather than throw away and make fertilizer? And the market output for avocado oils is probably 1,000 times higher than the fertilizer made from avocado. Just a question. I mean, there must be a reason why you chose to make fertilizer rather than make a product which can go to the market direct. Then Oliver, he was not very clear on his experimental designs, especially the in vivo. I would wish you tell us how you tested in vivo, so that you saw that the extract, the Wambuge extract, could control. So please expound a little bit so that we can find out whether it actually makes sense at this point. Then I forget the name of the lady we just presented. Pauline, yes. Pauline, yes, I want to imagine your choice of the source of the product or chia was informed by some previous studies, was it? No? Why then go for external, or for, if it was not based on any previous research, why not utilize very many means that we have around that are available to all farmers? Why are you going to, if a farmer deep in Meru, they won't even know what chia is. They can't, even if you put it in front, they won't tell you it's chia. There are so many other means. And I can give you an example. Leonotis, it's a big thing. It actually grows as tall as chia. It's a mint, locally available everywhere. And there are varieties of mints, including hiyo Kikuyu, maigoya. So unajua hiyo? It's a mint as well. So please, don't go and test things that are not going to be available to farmers to use. There are very many mints you can use if you really want to use mints. And you are likely actually to get, probably, I'm guessing, from the more wilder plants are likely to have the compounds you're looking for. Thank you very much. Those are the only questions I have.
Anyone else? Yes. Raphael? Oh, you already have the mic. Okay. Ask only 3 questions, no more than 3, and yes. Quick, short.
They are short questions. One to Lawrence Kinoti. Project, this is on logging moratorium. I was asking, how did logging moratorium impact climate change? Yes. And to Victor Kipto, in your diagrams on yields, you told us farmers who use DAP with CAN got more yields. Then why should we use other means yet we can get good yields, better yields with DAP and CAN?
Okay, Kirui.
Okay, thank you, Chairman. Short question to Purity Wanja on the drivers of water pollution along Kuru River, a tributary of Tana River. You talked about the water quality, and I was wondering if you could tell us what is the good quality of that water we want to achieve. I thought to talk about the physical, chemical, microbial qualities of water. And you talked about the collection of samples along the river. So I was wondering at what intervals. That one was not very clear. And finally, the indicators of polluted water. I think that one should come out very clear. Sharon, on the potential of black soldier fly larvae to recycle different organic waste streams into competent organic fertilizer. Competent organic fertilizer is the key thing here. I really wanted to understand, what is this competent organic fertilizer? What does it have that is so different to make it competent? Jared, on camel milk, you told us earlier that it is alkaline, and I'm questioning if it is alkaline. Is it healthy for, is it good for human health? Or something should come out clear from there. And Oliver, the last one, Oliver, on the biological control of bacterial wilt. You concentrated more with the stem pack and the leaf crude extracts. There is also another part of the Wampugia ugandensis, the fruit. Are you leaving out the fruit, or is there a possibility that it can also be utilized? Thank you.
Thank you. Anybody else? Njoka, you want to come back again?
My question is the person dealing with the avocados. I beg to reject that it's not all avocados originate from Central America. Rather than, I think it's good to see the avocados that are growing locally. It doesn't mean that they are fuerte or they are us just by looking. Maybe a taxonomy could be done. If they're not in the papers,
Raphael, what is your question? You are giving a speech.
My question, it's a comment I heard that all the avocados are from Central America. I beg to differ with that because we have local varieties which have been growing and they are doing good.
You are not the presenter. You are supposed to ask a question.
Okay. Okay, fine. My question is directed to the last presenter who is dealing with chia extract to treat the pest in cowpeas. I was asking the economic aspect of it. Is it viable to use the chia extracts to treat the pest in cowpeas? Because cowpeas are low value crop.
Thank you for your question. Okay, Njoka, the last one. And you can also ask each other, it's okay. Even if you're here, you can also
For
ask.
Alex Kimuya. Yes, the first question is, what are the specific limitations or the challenges which are associated with the current detection method for cooking fuels? What are the challenges? Then for this system that you have brought, what are these performance metrics or those indicators which you will use to evaluate the effectiveness or the effectiveness or the accuracy of the sensor system? And finally, what limitations or challenges do you think will occur during the development or the implementation of the sensor system? Thank you.
Thank you, Njoka. And now I have a couple of questions, starting with Alex. This question is for Alex from Dr. Cynthia Mugo. What you have developed shows very good accuracy, but how about the sensitivity of the model? Because some pollutants are normally at very low concentrations, and also the particles are of different sizes. So she wants to know about the sensitivity of that model. Do you have data showing sensitivity and the type of pollutant size-wise that can be detected by your model? I have a question for Kenoti about the logging moratorium. You said you chose 2 CFAs, I think purposely, and you didn't say how many, from what population. You chose 2 CFAs from how many, from a population of how many? And I thought you said something to the effect that you did that because of money, whatever, those kind of issues. And I'm wondering, for a questionnaire, how does the question of cost come in, even if you use a different method? I have a question for Purity, but I think it has been asked. You talked about the quality parameters from River Kuro, and I just wondered, why did you just focus on physical-chemical and not microbiological qualities of the water samples? Victor, I had a question for you, but I think it has been asked, so I leave it. For Sharon, I just wondered, you said you chose the BSF, and you want to find out whether they can biodegrade the avocado waste. Do you have a hypothesis? Is there any reason why you are concerned that they may not be able to biodegrade avocado waste? From the literature or from the knowledge that you have? For Jared, this is just a simple one. You said for making cream from chamomile, you did it at 50 degrees centigrade. Is that from a published method, or is it your own creation, the temperature? Thank you. So I think now we can go to the answers. Has anybody not been asked a question? Oh, everybody has been asked. You can start from there.
Okay, thank you. I think my question is about the health, whether the butter is healthy. As compared to the butter that is already in the market from the cow's milk, we can say that the camel milk has unsaturated fatty acids, a lot of them, so it's healthy. Then for the temperatures of churning, there are already some research that have been done at those temperatures, so it's already a cited reference. Thank you.
Okay, thank you. I have 4 questions, and the first one is what is good quality water? Okay, thank you. In my presentation, it's only because we are constrained with the time, I had a slide that was showing the international standards that have been set by different organizations. That is WHO, IAND, NEMA, and USEPA, and CAPES also. So those are the indicators. If the physical chemical parameters that were being mentioned, if they are above the set standard, then we can say that water is not good quality. Then on the intervals, Kuru River has got 3 zones. We have the forested, that is the upper zone. We've got the middle zone, and we have the lower zone. For the upper zone, I chose 3 sites. For the lower zone, I took 3, and then for the lower, I also took 3 sites. And then indicators of polluted water. The physical chemical parameters were measured and then compared with the international standards. Yes. Then the last one, it's on the why did I choose only the physical chemical parameters? What I can say, I had so many other parameters that I could have looked into, but due to financial constraints, I had to go for the few. Thank you very much.
Okay, thank you so much. I'm supposed to answer 2 questions. I will start with the final one that was touching on the number of CFAs. I picked 2 CFAs from 9 CFAs in Meru County. And where I was talking of the limitation of funding is for the case that I said I used Meru County State Forest to represent now the rest of the forest in the entire nation. And even if it was questionnaire, going to administer questionnaire all over the country, it is an expensive affair. That is why I was touching on the limitation of funding. And for these two CFAs, they were picked from the 9 based on, first of all, the level of activeness or the number of activities. A CFA like Mutiene is known to be very active, and then I picked the other one that they are not that active to act as a control of the other one. On the impact of moratorium, on how it has impacted on climate change, in my findings, we have seen that when the forests were closed for tree harvesting, the sawmillers and other wood users reverted to the extension forest that is in the community. And in the solutions of solving the climate change crisis, in line with nature-based solution, we usually say we promote agroforestry and we promote more tree planting. So we ended up losing a lot of trees in the extension area. And when we talk of increasing forest cover to absorb carbon, we cannot do much into the gazetted forest because that area is already forested. We are supposed to increase the activities of tree planting in the extension area, which, as a result of moratorium, we lost a lot of tree cover. Thank you so much.
Good evening again. I have about 5 questions. And the first one is the risk of the black soldier fly larvae. I'm going to say as of now, and the research I've done, it doesn't have any risk against humans because the feeding stage is at a larval stage. When they go to the adult stage where they're able to move around, they do not affect anyone. They just look for a place to mate where the life cycle keeps going on. So it's no risk to human. The second question is on the avocado, organic and indigenous. We actually said it originated in Central and South America. And any other variety you may see is actually has already originated from there. So that's what we wanted to say. And when we say organic, we mean it has been grown without chemicals. So that is what we meant. About the competent organic fertilizer is we wanted a fertilizer that can actually stand against any other fertilizer already in the market. So it has to be, those particular standards should be equal to the particular fertilizer we actually give. And the last question is why we chose to degrade the avocado waste. I am currently in a living lab kind of setup in a group project where we are working on avocados, reducing avocado waste. So somebody else is already working on the products of avocado, but we also need to reduce waste all around, which is why I'm working on making a organic fertilizer. And the last one is why we chose to, if there's any doubt that it will biodegrade, there's no doubt it will biodegrade, but we're actually thinking at it from the version of, if we break down the avocado waste, we can use these particular components to actually grow something else. For example, an avocado seedling, it will probably grow better and it will actually be of benefit to everyone. Thank you.
Okay. Thank you. I have some questions to respond. The first question was the economic viability of biochar to the farmers. I can say that the farmer only needs a metallic drum with a lid and a firewood, nothing more. For the raw materials, it is either sawdust or the dust from, what do we call, sorghum or millet. You can pan in absence of oxygen and you get your biochar, or rice husk. Next question was on how much PSF to use to maximize the yield. From my findings, I used the 8.5 tons per hectare, which was almost the same as the recommended chemical fertilizer, that is the DAP, which is normally recommended by the fertilizer manufacturers. So for me, I can say 8.5, but more research needs to be done to find the optimum rate or the optimum quantity of black soldier fly or frass manure per hectare to maximize the yield. Another question was, what makes a virgin land more productive? Even though this is, was not part of what I was explaining, but I can help. The virgin land, you know, it has been pale land, virgin, more organic matter has been decomposing, but no exhaustion of nutrients. So once you come and till the land and you plant, the nutrients are high if you compare with the place which is not virgin. The place which is not virgin, which has been under production, there has been exhaustion of soil nutrients compared to the virgin land. Another question was on why PSF. I think it has been answered by my colleague. The role of trichoderma and not any other fungi. I choose to opt for trichoderma aspergillum because that one is readily in the market and it is being sold by the Real IBM and Dudutech. So I opt for that because it was easy to obtain rather than going to do sampling in the farms and do isolation. So it was more bit economical to me to work on it. Another question was, why DAP fertilizer for maize and not NPK? Nowadays we have different fertilizers in the market. Yes, and these different fertilizers, they are used differently depending with the regions. And I opt for DAP because it is commonly used countrywide. And what the crop needs is only nitrogen, phosphorus, and potassium. Potassium normally is always available in the soil. So I opt for DAP because it has nitrogen and phosphorus. And I top up with CAN because to increase more nitrogen availability. Another question was yields. Why use other means yet we get good yields on? Okay, sorry, I missed that. The role of the trichoderma was it helps to solubilize the nutrient in the soil. It fixes the immobile nutrients to mobile, which is to be available for the plants. Have I answered? Yes. Unless you help me because I'm not a biologist. Okay. Okay, thank you. Okay, thank you. Unless now. Yes, I think I can help there because we are in a world where all people everybody want to sell what he has. And if I go to him and explain to him why DAP, at the end he will accept and leave NPK. It was on a light note, Dr. So DAP is still, in our region, we use DAP. The last question was on yield. Why use other means, yet we can still get the same yield using DAP? What I can say is that it is because of climate change. And here, when my colleagues were discussing about the conservation agriculture, we are trying to restore the soil health. And the more we pump in the chemicals into the soil, we are reducing the beneficial microorganisms which help us in fixing some of the soil nutrients in the soil. So one way of restoring this soil health is using organic manures. Thank you.
Very brief, okay, I was asked 2 questions about the in vitro work. In vitro work, I carried out using agar-well diffusion method, where I used tetrazolium chloride medium, and in that, in vivo, after in vitro work, the concentration that gave higher inhibition is the one which I carried to the feed, where I used soil drenching method in a complete random design. Then the second question was I concentrated on the bark and leaf. Why not fruits? Maybe that is a research to be carried out in the future.
Yeah, thank you. I have 2 questions, one from Moses. He had quoted the first abstract that we submitted here. Unfortunately, through the development of the research, we shifted from the use of cooking fuel as our study sample to the data from the environment for some technicalities. But on the same, I'll answer him 2 questions in substance. The benefits of using these evaluation matrices or metrics in our model's performance. It's always good to be able to tell from a statistical perspective how best your model can track the patterns between the given inputs and that what it can predict. And we don't use the eye to do that. We do some statistics. One of the measures we use is what I projected, the R-square score, and if it ranges between 0 and 1, then you're sure you have that score. So what matters is how higher is that value. But R-square score alone cannot be used to make a conclusion, so we need to use other common measures like root mean square errors and absolute mean square errors. their values should be very low when R square score is high, and that is what I projected based on the performance of our models. And since it's kind of quite a new idea in that field, we had to use those common measures. So the benefit is many people or researchers will understand the techniques we used in evaluating our model. Actually, they are very suitable techniques for evaluation, for that matter. The next question was on limitations of developing the sensor system. Of course, when you go to our local markets, you'll find these many sensors. But from the word sensor, you can be assured that a component that you buy is doing some kind of sensing from the environment. And it might be limited, so it might not have the capability to measure all the parameters you might need. So one limitation is we had to hook up so many sensors to acquire the desired study data. Very good questions from Dr. Cynthia. One was on sensitivity of the models. And I think here I will answer this question in terms of the particle size, because this seems to connect to the second perspective. We are dealing specifically with particulate matter pollutants, and the current classification puts them into 3 categories. We have P1.0, P1.25, and P110. The existing techniques for detecting these PM concentrations have serious limitations, and one of them is sorry. Okay, so let me finish.
Briefly.
Yeah. So the question was, how did we ensure sensitivity? The sensor that we used has no issues to do with the sensitivity of the particles because it is genetically designed to measure those 3 parameters we are dealing with. And the prediction models have to rely on the actual data that we collected using this sensor to make predictions. So we are confident with our output. Sensitivity and types of pollutants that can be detected by the model, we are talking about 3 as of now, but the future allows us to extend to other types of pollutants that exist in the atmosphere.
Okay, quickly, the first question was why not utilize other means? Actually, also a comment. I take that in stride, and maybe I can think more on that. But listening to that question also, a song comes to mind, Vunja Mifupa, Kama Bado Meno Iko. We look at the relevance of chia right now, and it has been hyped in terms of the chia seed, but we are looking at these other parts like the leaves and the root bark, and we're trying to see a kind of value addition to see what else the farmer can get from this plant. In terms of, we are using a high value plant to control pests in a low value, something that is considered a low value crop. Colossobruchus maculatus, or the cowpea weevil, is not only limited to the cowpea. It's not a pest only to the cowpea. It attacks most of the legumes in the legume family. Number 2, as I had said earlier, we are using not the overhyped chia seed. Even though we'll subject it to the experiment, our main aim is actually the leaves and the root bark. And of course, we are looking at growing the knowledge and of course pushing the envelope. And if it works on the cowpea bruchid, of course it will also work probably on other pests as well, because it cannot be specific to the cowpea bruchid. So you can subject them to other insects that we term pests to more economically viable crops.
Okay, we've come to the end of our presentations. Everybody, let's appreciate all the presenters, please. Let's appreciate them. It's been quite a marathon for all of us. I want to thank you for the patience that we have all exhibited and the commitment to add value to these presentations, not worrying about being late. That's very commendable. We appreciate that. We go again tomorrow. We have more presentations tomorrow and the day after, and I want to ask you to kindly help us to spread the word so that the hall is full. We want real attendance so that the presentations can all be critiqued properly, and we add value to them. Okay, now I'd like us to conclude, and I would like to ask somebody to, after my colleague here makes a final comment, maybe, I would like to ask a volunteer to pray for us, and possibly, if you haven't spoken today, please, I would like you to be the volunteer for that. Okay.
Thank you, Chair, for taking us through that session in a very nice way. Thank you very much, staff audience, especially Dr. Mugambi, who was chairing the earlier session, has been here throughout. I saw a few others. Professor Moria just walked out, Dr. Cynthia, and a number of them. Thank you. You can see, they say, if you want others to listen to you, you have to listen to them. And I think we have observed that spirit. We were here to listen to all our colleagues, and they were here to listen to us. I think that is really the spirit in which we do a conference. Remember, about conference is also about networking. If you have left today, well, he has said that without having spoken, I will add to, without having gotten a phone number. A lot of your presentations were getting into each other. You can, even from yourselves, you can actually add value to what you are doing, especially those who are still doing concept notes, proposals. It's very important. So please do that. Of course, even tomorrow and so on. The other comment I have to make here, I don't want to embarrass anybody. There's some work that 5 years ago, those topics were still being presented here. I don't know if the head is in sight. This work has been going on since when I can remember. I've seen some people in biological sciences lab since I can't remember when. So please, guys, we want to end that trend of research forever, because the nature of research is that it gets overtaken by events if you delay too much. Okay? So those who are starting, have the end well in sight, do it in time. For masters, you know, it's 1 year coursework. The other year, in 6 months, you should be through. Maybe before you graduate, another year ends, and that's it. If you want to continue, there is a PhD, or you can do a second master's, and so on and so forth. So with those few remarks, I think I want to end the comments there. Thank you very much once again for your attention, for your cooperation, and for everything. Let's continue like this until the end. It is not over until it is totally over, or all over. Thank you, Chair.
Thank you, Dr. for those encouraging comments to our students, postgraduate students. Actually, we keep preaching that we want you to graduate within 2 years, because an MSc is a 2-year course, a PhD is a 3-year course. If you extend it so much, certainly not more than 3 years. So, those of us who are still starting, make sure that you do your best to graduate within the required time. Thank you. Let's stand up, and may I have a volunteer to pray for us, please?
Okay, let's pray. Father, in the name of Jesus, we are grateful for giving us a good day. Thank you for successfully seeing us through, and for enabling each of us to participate and to get involved as they should. We pray as we go that we enjoy journey masses, and by tomorrow we pray for more grace to finish up this activity in good faith. And it is in the name of Jesus we pray and give thanks. Amen.